Well, hello, everybody. It's excellent to be with you today. I was appreciative that Dr. Jelen invited me to speak to you on this day of consumption of quinoa. I am going to talk a little bit more on the molecular side and what we're doing to try to improve quinoa so that we can have more consumption in the future. So let me just slip over to my slides here. I'm going to share with you my presentation, and I'll go into full mode so you guys can see that there. I'm at Brigham Young University. Topic here is just genomic resources. These are the resources that breeders use to improve quinoa. Hopefully that leads to more production, more consumption. My colleagues are Rick Jelen, David Jarvis. Lots of people worked on this with me. I certainly don't want to suggest that I'm the only one working on quinoa at BYU. So I just want to bring their names into this. We're talking about the amaranths. I just remind you guys, there's a lot of different species in the Amaranthaceae, and many of them are very important, like sugar beet here, or spinach, amaranth, of course, and even atriplex. We've done genetic resources for all of these different species. We're focusing in today on quinoa, which is also part of this family of Amaranthaceae. So just a quick update, Dr. Jarvis will talk to you a little bit more about what's going on in C. quinoa. We had an original publication for C. quinoa that showed us the basic outline of the genome. And I'm now, because the last time I talked, we didn't have it available, it is now available for people to look at, to utilize, which is a chromosome scale assembly that we've done in collaboration with Mark Tester's group out of King Abdullah Science and Technology University there. Very excited to have that resource available. Again, tune into Dr. Jelen or Dr. Jarvis's talk to you a little bit more about that. I'm going to talk to you more about the effort to make resources available for production of quinoa in the United States and elsewhere where it's not adapted. What we think will have to happen is we're going to have to use extant resources or species that are present in the United States or in Europe to cross in genes that will be important for biotic resistance or abiotic resistance. But you got to know a little bit more about quinoa to understand why we're doing this and why we think this is the case. What you know about, of course, is quinoa, which is derived, we believe, from a weedy species called C. hircinum, which itself is derived from another weedy species called C. berlandieri. You'll note that C. hircinum is found in South America. It kind of grows in a crop weed complex, and that C. berlandieri is actually from North America. Indeed, we believe that the original origins, not the domestication, but the origins of the species themselves are coming way back here, probably into North America. Many reasons to believe that's the case. And this C. berlandieri is actually the hybrid between two different plants, one of which was Eurasian, the other which was North American at that time. We call this the A genome and the B genome. They come together to make what we now currently have as C. quinoa, which was domesticated probably in pre-Incan times around Lake Titicaca. What we've done is we've sequenced quinoa, but we've also gone and sequenced what we think were the original two parents way back here. We're talking millenniums, millions of years ago was this probable hybridization occurring. We chose, obviously, these plants no longer exist. They've changed through evolutionary time, but we've chose something that's a representative of them, that's extant today living. And what we picked was the A genome C. politicale. And we, of course, picked that one, not because we think it's the closest one to this original parent, but because it has other uses. It's also a superfood called quinoa or quinoa, depending on which area of South America you're from here. So very important one. And then C. suessicum here, which we think is a representative of the B genomes. Again, we had very little information when we were starting this. This is older work from 2017. Doesn't seem like it's that old, but it certainly is at this point in time, the speed at which genomics is moving. And just from this little bit of information, we could determine who was the original maternal parent, and it likely was the A genome, and we represent that as politicale here. So we're starting to get a sense of the evolutionary history of how this works. I just, again, to remind anybody watching this, if you're interested in having the information about quinoa or quinoa, this information is all publicly available. And I just want to make a call out to my colleagues that helped us do the quinoa work that allowed us to collect, utilize DNA, build our first genomes. Again, remember, this is an A genome. So when we look at politically relative to the A and the B genomes of quinoa, what you see is it's much more related to the A genome, just like you would expect. Once we had this information, we started to ask ourselves, how closely related is politically to the A genome of quinoa? And it turns out that there's other species that are actually closer. Not surprising, I guess, in that sense. And we've sequenced some of those. This is the C. watsonii genome. We're very excited about it, once again, because we saw that it was much more related to the A genome of quinoa. And of course, again, this species is in North America. This species, politically, is in South America. That's why we believe that we had this original tetraploidization event, this hybridization event occurring in North America. So we have sequenced that, but it really made us think, do we really know? Is C. watsonii the closest one? You don't know until you start to look at all the different diploids that are present in the North American collections. And we had to actually go out and do most of these collections ourselves. But you can see there's just a ton of different diploid chenopods out there that could be the A genome parent in those cases. And of course, we didn't limit ourselves to just North America. We went down to South America to look there as well. And we have some exciting stuff that comes out of that. But what can you do with these diploids? You can start asking yourself, do they make sense? And what I wanted to show you guys here is the ones in blue are all the A genome diploids, very different when you look at their sequences relative to the B genome diploids. These would all be from Eurasia. In fact, this Borbishai has now been renamed into Ukrainian. This Sergey was just telling me about that. So these are all B genomes, these are all A genomes, and then this Volvaria is kind of our outset. It's an H genome, so it's something way out there in the middle. You can kind of see nicely separated. So we now have a good set of resources, not just one or two A genomes. We have multiple A genomes and multiple B genomes for that much. So how can we use this information to get a better sense of what the true parent was, at least from the North American diploids for the A genome diploid? Well, all we had to do was tease out what are the A genome chromosomes from quinoa and compare those A genome chromosomes to these A genome diploids. And when you do that, remember we said watsonii is closer than is politicale, which it appears to be. But when you do that and you look at the A genome from QQ74, which is quinoa, it actually is not in with watsonii. So we wasted a little bit of time sequencing watsonii. Politicale was very important, of course, because it's economically valuable, but look where it falls out in the leptophyllum. If you'll think about the map of the United States, leptophyllum's there in kind of the southwest portion of the United States. So we're excited now to start saying to ourselves, we think we know what the true diploid parent is there. You can do the same thing on the back, You can do the same thing on the B genome. And remember what we did is we sequenced Suassicum, but it turns out that Ficifolium is probably more similar to it. And so we're getting a much better sense as we go forward in these different lines out here. So then we kind of ask ourselves a different question. Can we improve quinoa with these existing tetraploids? You wouldn't cross through the diploids that I've just been talking to you because you'd make a triploid, which would be sterile. But we have lots of tetraploids that are out there that we've also collected throughout the United States and Mexico and South America. And you can just see some of them. They are called C. berlandieri. Remember, quinoa is C. quinoa, but C. berlandieri has a lot of different subspecies or varieties associated with them. In fact, one of the most important ones we found here in Mexico called C. berlandieri subspecies nuttaliae, which is called Mexican quinoa. Oh, okay. Huasantlequias, chilites, depending on how it was used. Sometimes they use the leaf, sometimes they use the actual inflorescence, other times they use the grain, like quinoa is used for grain. So different, if you would think about it, accessions or ecotypes that are present there. So we've gone ahead and collected those with some help from a colleague, Eulogio de la Torres in Mexico. And we've had a lot of success now developing a brand new assembly of Mexican quinoa. So we're really building out these resources, but there's lots of other things out there. And I'm not suggesting that C. quinoa as we know it, came from nuttaliae, probably didn't. And I'll explain why we think this. They were probably separate domestication events by different civilizations. If we look at what's happening up here, is we have all these different varietal types of berlandieri, macrocalicium. We're excited about that because it has high salt tolerance, and Bocianum down here right along the Gulf. Again, would have it's growing out there in these tidal marshlands, seawater coming in, probably has tremendous heat tolerance, probably has tremendous insect tolerance and salt tolerance. How about drought tolerance for the sinuatum or Shiitake? So we're very interested in those and asking ourselves, could we cross C. quinoa to one of these tetraploids and move some of these genes into quinoa so that they're more adapted to production in the United States or into a local environment? Again, these plants have been growing in these local environments. They have resistance genes. They have tolerance genes already for those localities. Can we move those into C. quinoa and grow quinoa in these locations? And so again, I would show you, here's quinoa. And there's some diversity in quinoa, as one knows. There's these lines here that we think of as being, coastal types. They're very different. Again, those might have actually been separate domestications of C. quinoa or C. hircinum. There's some argument back and forth, perhaps by ancient tribes, ancient peoples that were living along the coastal region, the Mapuche or somebody like that. And then, of course, you also have pre-Incan civilizations domesticated in the Altiplano, and we see these high Andean Altiplanos. You also have the valley types and others. But here we have quinoas. Here we have the ancestors, the relatives, the tetraploid relatives from the New World. And of course, we're very interested in, say, like nuttaliae again. Nuttaliae is Mexican quinoa. Can you make crosses? I think there's been people that have suggested this, Alejandro Bonifacio for one. Others have made this. What we want to do is prove this from a molecular stance, not just have phenotypic, but have genetic molecular evidence of this. And I just wanted to show you an example of Bocianum. This is what we call BYU 937 Fat Boy, because it was growing on a dock near a restaurant called Fat Boy there. So we've collected all these, and now we're resequencing these. And we've sequenced now berlandieri, nuttaliae, complete. Again, it looks a little bit different than what you're used to as C. quinoa because it's used for different reasons. In this case, could have been used for the inflorescence. So in our science paper, we made this kind of suggestion that you had movement of these ancestral weedy species that were the tetraploids down somehow into the Lake Titicaca regions. I think perhaps this is misleading. If I look at bird migrations, almost all of them go down, this stretch of land where, we have this connection between, the wild ancestors, the weedy species. My guess is, since we believe it's probably delivered through bird migrations, is that you have this large migration coming down this peninsula here, this stretch of land through the Yucatan Peninsula, and then coming back into what we think of as South America. That suggests something to me. That suggests that if indeed you had the tetraploidization event occurring here, then you have some plants being domesticated here by pre-Aztec and pre-Mayan peoples, probably here. Again, these are very important, continue to be regionally important crops here. And then you also have quinoa here. If there was one species that's coming down through here and then evolving and then being selected from and domesticated, it should represent a single domestication event. And when we look at that, we can actually show that indeed it was a single hybridization between two different diploids that led to all these different tetraploids. And again, all we've done here is we've tried to look at, here's the A subgenome for all the different tetraploids. Remember, this is Mexican quinoa here. Here's quinoa. Here's hircinum that we think we got it from. They all are derived from the exact same tree here, suggesting once again, these things came from a single, or at least the same parent that was the diploid parent. That was the A genome parent. And then when we look on the other side, same thing happens. It's not like they're going to different, it's not like nuttaliae, which is Mexican quinoa, is out here with hyans, and that quinoa is out here with, petiolare or something like that. They're all coming with the same one. And indeed, what you'll notice here is they're all coming from that same leptophyllum group that we're seeing or we're thinking is happening in these cases. And the same thing is going to be happening over here. They're all coming from that same Ficifolium group that we have in that sense. So again, probably a single domestic, a single tetraploidization event, spread of the wheat, more likely coming down through Mexico, down peninsula into Colombia, and the wheat is just moving and then becomes specialized and domesticated by these different populations that we have there. And I think we have pretty good evidence of that at this point in time. So are the wide populations possible now that we know something more about these? Can we make crosses with these things? And indeed you can. Now, we've made our crosses passively, meaning we've just put the two plants together. So here's Real, high altiplano type, quinoa, and here's Bocianum. This is from the Gulf states, from Louisiana. And we simply put them together in a tent, increase heat that tends to kill the pollen in the Real, in the sea quinoa, but doesn't affect, because again, there's heat tolerance in the Bocianum. And then we look for crosses. So we're thinking that pollen is coming from here, pollen is dead here potentially, and that we're getting crosses up here. Of course, when you harvest the seed off of here, they all have the maternal look. They all look white. But when you grow them up, you see what looks like Real plants, and then every now and then you see this crazy looking plant, which is probably the F1 hybrid. And we've always assumed that, but now we really wanted to put, molecular data to this to prove this. So here's what one of our crosses looks like. This is a Hercinum, this is Real, and once we get something we think is a punitive F1, it produces seed that looks like this. It's kind of an in-between. This is a quantitative trait. And at the same time, we can ask, when we look at this F1, we can ask some questions. Does it have, it's obviously black, but does it have the chloroplasts, which are different between these two? Does it have the chloroplasts from Real? And it does. And that's what we're showing you here. What we have, in essence, is we've known this. Most plants show maternal, uniparental, maternal inheritance of the organelles, the mitochondria, the chloroplast. And we've shown that here for both mitochondria and chloroplasts. Quinoa is, as probably expected, but now proven, shows extranuclear organelle inheritance through the maternal parent. So we now have taken these crosses that we've made. We made crosses through Hercinum, which is much closer. And then we've made crosses to Bocianum, which is much further away. And here's the population of Bocianum. This is the F2 seed. And you guys can see very different, has very different looks to the seed heads. I'll show you some pictures in a minute. Has very different germination rates, but they're viable. These are true hybrids. And the reason I can tell you that they're true hybrids, let me just show you the next pictures. They're producing seed. You guys can see the seed down here. These are definitely, making hybrids, and we're making seed. And I show you guys here linkage maps. This is between C. quinoa and C. Hercinum. And this is between quinoa and C. Bocianum. What a linkage map shows is that there's recombination occurring between the chromosomes. So we have a quinoa chromosome now with the Bocianum chromosome. They're aligning it, they're synapsing during meiosis, and they're exchanging genetic information, which means we can grab genes from these wild parents and push them in. So one of the things we wanted to prove is, one, can we make the hybrid? Two, is there recombination occurring? And then three, can we use these to map things that would be of interest to us? The wild varieties oftentimes have a stinky fish smell that comes, it's derived from trimethylamine. These trimethylamines is a volatile organic compound. It's released probably by the plant for a number of reasons. It's probably an attractor and brings in insects or other organisms to help distribute its seed. Whereas quinoa doesn't need that, so it's been selected against. And obviously, we don't want to move a smelly, gene into quinoa. And so we'd like to, if we're going to make crosses with the white stuff, we're going to have to select against that smelly gene. So just as a proof of example, what we're doing is we're now mapping that. And so we have 937 This is done with a number of people, Sean Christensen and Jeremy Winders with the USDA in Gainesville, Florida, where they're using this proton mass spec, and they're able to then distinguish the levels of the smell. You would think, well, Jeff, couldn't you just smell the individual plants and you would either smell or doesn't smell? It's probably a single trait, but it's probably quantitative and your senses get overwhelmed quickly. So we're having to use, sophisticated devices to actually get these quantitative measures for this. So our hope is that in these quinoa by Bassianum crosses, we'll be able to map where this gene is and then specifically, select against that region of the genome as we move genes in. We're very excited about this. And I've already shown you that it looks like the crosses are real. We have the genetic data to show that we have recombination blocks coming from quinoa, recombination blocks coming. So back crossing, we can really move in resistance genes into these different species or different characteristics like heat or drought. Now, the last thing I wanted to just say is that we're very excited now to actually have the first hexaploid fully sequenced to the chromosome level. Now, you'll remember the B genome is from Eurasia, so are the C and the D. And we were very concerned that the C and the D would be incredibly close and we couldn't separate them out. But using PacBio sequencing data, we were able to identify each and every one of the chromosomes specifically. And because we had satellite or repetitive DNA sequences that were unique to the C and unique to the B genomes, we were able to identify the exact chromosomes to their subgenomes. So we know these are the B subgenomes. They're fully sequenced, telomere to telomere. Some of them indeed go across the centromere, which is very exciting and isn't done in very many plants. And then, of course, we also know not only the B and the C, but also the D, and we were able to distinguish between this. This has been a wonderful collaboration with a group of scientists in Taiwan. And the reason we picked Chenopodium formosanum instead of the weed, which might be argued as the more important because it agriculturally is very destructive, we picked this BCD form because it's now another up and coming superfood called the Julis or Honghui, and has an ancient history, just like quinoa, for the Taiwanese people. So we're excited about having this new resource. The last thing we're working on now is a strictum, Chenopodium strictum, which is the tetraploid that hybridized with the B genome diploid to create what we think of as the album complex. So very exciting new tools coming forward, and we're excited to share this with everybody. If you have any questions about this presentation, or if you want resources from this, don't hesitate to reach out to us. Thank you.